I'm working in Professor Ivan Good's lab, and he's looking at the mTOR pathway and all its signaling and all the components of the pathway. So we have already established like stable human kidney cells that express this mutant protein. So what I'm trying to do is essentially like replicate the same process but in mouse cells. A stable cell line is just cells that have taken up the gene and are manufacturing proteins that are functional and stable. So basically what I'm doing is I'm culturing cells that express my protein, and after that I'm taking these cells and running a Western blot to make sure that they manufacture the protein I'm looking at. So here I'm taking out cells from the incubator, which leaves them at a temperature of 27 degrees and with high humidity and high CO2 concentration. Here I'm checking the flask to make sure that there isn't any cloudiness, which indicates contamination. So I'm taking out the medium from the water bath where I preheated it. And before I put anything into the hood, I have to sterilize them with 70% ethanol. So here I'm removing the old medium and throwing it away. So here I'm adding trypsin, which will help my cells to detach from the walls of the flask. So I collect the trypsinized cells in a falcon tube and centrifuge them to pellet down the cells at the bottom. I spin the cells at 2000 RPM in the centrifuge for 2 minutes, and I have to make sure that the centrifuge is properly balanced before I use it. So here now I can see that the cell pellet has collected at the bottom, and I'm now taking out the supernatant at the top. Here I'm resuspending the cells in media again to break them up. Here now I'm adding new fresh media into the 6-well plate and the small flask that I'm using to subculture my cells in. So here now I'm adding my resuspended cells into the flask and the plate. At the end of every session, we have to make sure that the hood remains extremely clean, and we would do it by sterilizing the working bench using ethanol and closing it and switching on the UV light. So I will collect cells from my tissue culturing and I will lyse them and use them in my Western blot. So I'm using the technique of Western blotting, which sort of separates the proteins in my samples according to their size on a gel and transferring these proteins onto a membrane. With this membrane, I'm incubating it with a primary antibody that binds to the protein that I'm looking for. And I'll be using a secondary antibody to bind to this primary antibody to sort of show a signal as to where my protein is on the membrane itself. So here I'm trying to find out the location of my protein by incubating the membrane with some chemiluminescent substrates. The protein then will be visible on the screen as it exhibits a chemiluminescent signal. Here you can see the band showing the protein I have successfully expressed in the mouse cells in the tissue culturing. So I think like some of the skills I've picked up through like working in the lab is that I have to be like really organized, and I have to sort of note everything down in a notebook and sort of plan everything really well so that I know when and like where I'm supposed to be. So what I've enjoyed most about the project is actually working with the people in the lab, and it's quite nice like how the way they treat you, because I think like learning in the lab is sort of a one-to-one, it's a hands-on thing, so it's not something you can actually learn about in like a lecture, and it's more like personal, and that's why I quite enjoy working in the lab.